So, what's the deal with chromatin? Basically, chromatin is DNA that's been wound and compressed very tightly. DNA is incredibly long, and in order to fit a full copy of your genetic code into the nucleus of every cell in your body, it needs to organize itself into this tightly packed form. If you unwound all of the DNA in a single one of your cells, it would be about as long as the average human wingspan. And if you unwound the DNA out of every cell in your body, it would reach all the way to the moon. So how does all of that fit into the teeny tiny nuclei of your cells? Well, we're not just shoving spaghetti into a bag here. This stuff is really organized. Your double-stranded DNA helix winds itself around histone proteins to form little wound spindles called nucleosomes. These beads on a string condense to form the 30 nanometer strand, which supercoils around itself and can be held together with scaffold proteins to form intricate chromatin structures. These structures organize into various higher level structures, including topologically associating domains, chromatin subdomains, and the open and closed chromatin compartments. All of this comes together to form the dense, organized mess of chromatin that is the nuclear genome. When our cells suddenly have the need for a protein that performs a particular function, chromatin rearranges itself so that the region of DNA containing the gene that encodes that protein can be accessed by the molecular machinery that converts DNA into proteins. And when certain proteins aren't needed, chromatin packs them tightly away for storage efficiency. But how efficient is this process of opening and closing chromatin for specific needs? It would be energetically favorable if genes were organized in 3D space, such that activating and deactivating the genes needed for a specific function were close together. But is that how it actually works? This brings us to our research question. Do functionally related genes cluster together in the nucleus? Specifically, the hypothesis we've been testing is this. We hypothesize that genes related by the same gene ontology term, metabolic pathway, or protein-protein interaction will significantly cluster in the nucleus. Furthermore, we hypothesize that the extent of this clustering varies between cell types. Since I belong to a bioinformatics lab, I've been tackling this problem with maths and coding. Our method is based in graph theory, which is a branch of mathematics inadvertently invented by Euler as he wrote to a friend about a way to cross 7 bridges in the historical town of Königsberg, Germany. Euler codified his solution such that locations that could be traveled to were represented by points, or nodes, and the bridges that connected them were represented by lines or edges between the nodes. These days, graph theory is used extensively in network analysis problems, including methods that analyze relationships in social networks and problems relating to biological networks. In order to apply graph theory to our chromatin organization problem, we're taking advantage of a technology called Chromatin Conformation Capture, or 3C. 3C is an umbrella term for a series of methods that work in more or less the same way, but at different scales. In these methods, chromatin is fixed in place using formaldehyde, and then chopped up into even pieces by a pair of molecular scissors called a restriction enzyme. These bits and pieces are ligated to the pieces around them in close proximity. This leaves us with hybrid DNA strands, where one half of the strand is DNA from one region of the genome, and the other half is a different DNA that was close by in 3D space. In the specific method we're using, called Hi-C, these hybrid DNA strands are put into a next-generation sequencer so that we can get upwards of 4 billion data points containing information saying this DNA was close to this DNA. Using these methods, I've created software to convert these Hi-C data points into a graph and then perform clustering analysis on that graph to see whether or not pieces of DNA annotated with specific function are found closer together to each other than would be expected by random chance. To accomplish this, we download Hi-C data from a publicly available repository, align it to the human genome, bin that genome into even chunks that we represent as individual nodes, and assign weights to the edges between those nodes based on the number of times we see the same two pieces of DNA show up together in the experiment. Then, we normalize the graph, annotate our nodes with gene functional information, and assess clustering based on those annotations. We use a measure called total pairwise distance to assess how close nodes in a given cluster are in relation to one another, generate significance values using a Monte Carlo sampling approach, and then perform multiple hypothesis testing correction using a permutation test. I'm currently working on testing different clustering measures in the software to see what gives us the best signal, and have some work to do on getting everything running smoothly, but early results have been positive. Although we are not yet able to effectively determine exactly which functions are being clustered together, we have been able to see that there is an effect of function on the extent of clustering between nodes. This figure shows one of the functional terms we looked at colored in red, very tightly clustered amongst nodes that do not have the same functional annotation. This is especially noteworthy considering these nodes represent genes that exist on separate chromosomes, but have seemingly been brought together in space regardless. In summary, we have created a network model of chromatin as it exists in 3D space, and are using it to look at how gene function relates to chromatin organization. Thanks for listening.